Okay, so in today's video we are going to discuss about cell wall differences. Like we have two type of differences. Cell wall differences, gram negative and gram positive. Gram negative, gram positive. Okay, so we'll just go with a flow chart to make you understand very easily. First thing we'll go with character. Then we'll go with gram positive. Then we'll go with gram negative. Okay. So how we'll go? First we'll start with outer membrane. When we talk outer membrane, so when we talk about in gram positive, so outer membrane is absent in this one. And when we talk about gram negative, so there is, so this means present of, in gram negative outer membrane is present. And when we talk about cell wall thickness, this is about outer membrane, here we have cell wall thickness. So here in this gram positive we have around 20 to 18 nanometer. This means this thick. And when we talk about this one, we have 8 to 10 nanometer thickness. So this is called as thin as compared to gram positive, right? And when we talk about peptidoglycan, this is main difference you'll find in picture 2. So thickness, not thickness but peptidoglycan layer. So we have like around, you might just, I will show you picture as well. So this is thick, this means multi-layered. This means a lot of peptidoglycan layers are present, multi-layered. And when we talk about this gram negative, so they have thin, so we just call this as a single layered. Okay, then when we talk about rigidity, so they are rigid enough. This means they're very hard, and they are less rigid. This means not very much strong, or we can also say if they are rigid, so you will also see that they are like, when we can also talk about elasticity. So in this one they are less elastic. If they're very strong, so this means they cannot move, they don't have much elasticity in their body or like in their wall, so they are less elastic. And they are more elastic. Okay, and we have other things too. Here we have variety of amino acid, like you'll see so many amino acids present, and so it's like they are telling we have variety of amino acid in which structure. Of course not in both, we have in both. Like here you will see few, which mean very less amino acid types, and here you will see several types of, so many, not so many but different type of amino acids. And you will also see they are aromatic, their ring like structure, sulfur containing amino acid is present. I'm writing, you'll see word which is aromatic, which means ring like structure, or it's like I'm just describing, this is a ring like structure which is connected with hydrocarbon, like they are linked with each other through hydrocarbon. Okay, So what they are telling here, they are telling that these are aromatic and sulfur containing, and that sulfur is even not presenting in so many, like in both, so that's why we have a difference. In this it would be absent, and in this one we'll be having, so it's like present and absent. I'm sure it would be clear to you. Now we have lipid and let me change color. Here we have lipid and lipoprotein content, lipid and lipoprotein. So here low acid bacteria have low lipid content, and here is high. And when we talk about, it's like only this is the difference. So, and let's go with the example so that you can easily identify. Here we have examples. Example in this one, first, here is positive. So gram positive have example of enterococcus, and bacillus and clostridium. Sorry for bad writing, I don't have enough space, so I'm writing down. And in negative we have E. coli as well, E. coli and Helicobacter. We have Pseudomonas, and we also have few other like Salmonella. We have so many examples, so I'm not writing all. So yes, this is all about gram positive and gram negative. I'm sure it would be clear to you. Thank you. Bye bye.